Megasporogenesis.
Megasporogenesis.
The definition.
The definition of megaspore. All of these things are clear to you, the things that we have discussed in the class?
Yes, ma'am.
Any doubts anywhere, like with respect to the structures of the megasporangia and everything? Any doubts?
No doubts, ma'am.
Very good. Now see, so in the previous class, we had discussed about the particular thing that is megasporogenesis. Now, what is megasporogenesis? It is the formation of megaspore, right? Megaspore from the megaspore mother cell, right? And this megaspore mother cell, imagine it as being, the initial cells to be making the megaspore. So the megaspore mother cell, it is differentiated by the ovule in the micropyle region. Is the micropyle region tip or the base? The micropyle region, is it a tip or the base?
The tip.
Very good. What is the name of the base? What is just opposite to micropyle?
Shalaz.
Very, very good. Yes. You're studying, huh? Very good. Yes. So chalazal end is the basal end or the base, and the tip or the top is the micropyle. Now, the megaspore mother cell, it is a large cell which contains a lot of cytoplasm. So a very dense cytoplasm and big showy nucleus is there. So they contain a prominent nucleus. Now the megaspore mother cell, it undergoes the process of meiosis so that they can form 4 haploid megaspores. So they form 4 haploid megaspores. Now, out of these 4 megaspores, only one is functional and the other 3, they are not functional, so they will degenerate. So the functional megaspore, it develops into the female gametophyte, that is the embryo sac. Okay, it will be developing into the female gametophyte, that is the embryo sac. And this, since only one megaspore is leading to the formation of the gametophyte, so we call it as monosporic development. Monosporic. Mono means one. Sporic refers to the one megaspore, and development you already know. So the method of the embryo sac formation from that one single functional megaspore, we call it as monosporic development. Clear?
Yes, ma'am.
Now comes the formation of the embryo sac. So now the nucleus of the functional megaspore, it starts dividing mitotically. Now can you tell me quickly what is the difference between mitosis and meiosis?
Mitosis is equational division and meiosis is reductional division.
So at the end of mitosis, what will happen? What will happen to the number of cells? Will they increase or decrease?
It will increase.
Very good. The number of cells will increase. And what will happen to the number of chromosomes, like the number of chromosomes in that cell? Increase or decrease?
Mitotic division, the chromosomes does not change the chromosome.
It doesn't change. And after meiosis, it reduces to half. Very good. So now, the megaspore will start dividing mitotically, meaning it will increase the number of cells. Nothing will happen to the chromosome number. Only the number of the cells will be increasing. So that from one cell, they can form 2 nuclei and that they will go to the opposite pole. Okay, so now the embryo sac will become 2 nucleated. Okay, then 2 more mitotic divisions will happen, leading to 4 nucleate and then 8 nucleate. So 2, 4, 8. Now total 8 nuclei are formed in the embryo sac. Okay, and if you remember from cell cycle chapter, that every nuclear division is followed by what is it followed by? Where did you go? The video is off. Every nuclear division is followed by the cellular division. Are you able to hear me? Yes or no? Yes, then? Okay. Yes, Justin, are you able to hear me?
Yes, ma'am.
Got disconnected.
Yeah, ma'am.
What is the last thing that you listened before the thing getting disconnected? Huh?
Um, Two mitotic divisions. You are in that point.
So after the two more mitotic divisions, so from the two nuclei, now four nuclei are formed, and then eight nuclei are formed in two. Now I said that every nuclear division, it is usually followed by the cellular division, meaning the division of the cell. But in the case of this kind of division, it is called as free nuclear division. Here, what happens is that the nuclear division is not followed by the cell wall formation or the cell division. Okay, so that now imagine this situation as that one cell is present and that one cell is containing all the eight nuclei. So it's like a multi-nucleated situation. Now, after this step, then the cell wall starts to form. Okay, and that becomes the female gametophyte. So now see, this is the whole entire thing that I said. It is mentioned in this diagram. So initially, it will begin with a diploid megaspore mother cell, and then it will be followed by meiosis and form the four haploid megaspores. Out of these four haploid megaspores, one will be functional and the other three will degenerate. Degenerate means die. The one functional megaspore will like do the process of mitosis and it will form two nuclei. Then it will further do two more divisions and ultimately form the eight nuclei. And then I said that the cell wall starts being laid up. Now let us see how that happens. See, out of all of the, you can say nuclei that are present in the cell, okay, there are eight nuclei. Six of them get surrounded by cell wall and they become cells. Six of them. And the other two, they are present right in the middle as polar nuclei. Okay, so see in the middle, this situation, this is the polar nuclei. They are not surrounded by any cell wall. Whether if you see over here, all of these have walls present. The red circles that you see, it's the nucleus, and the lines surrounding them is the cell wall. Clear? Now I'll explain this distribution. So for example, let's see the distribution. See, three cells, like out of the eight nuclei that are formed, right? We saw that six of them started to have the cell wall around them. So now the three cells, they move to the micropylar end and they become the egg apparatus. With what are the three cells? See this. 1, 2, 3. So these three cells together, it forms the egg apparatus. Okay. And you understand how it is a cell. This is a nucleus and this is the cell wall. So it becomes a cell. One cell, two cell and three cell. Clear? This arrangement is clear? So there are two synergids and one egg cell. So the two synergids and the one egg cell together, it forms the egg apparatus. And the synergid, it has the filiform apparatus. It will be helpful for guiding the pollen. You will see that later on. For now, just remember that at the end of the micropyle, there are three cells present and they make the egg apparatus. The egg apparatus consists of two synergids and one egg cell. Now, the synergids, they have special cellular thickenings, which is called as the filiform apparatus. It plays a very important role in guiding the pollen tube. We'll be discussing it later. Then there are three more cells that go to the basal end, that is the chalazal side, and they are called as the antipodal cells. These three together, this is the chalazal side, they are called as the antipodal cells. And then the large central cell has two polar nuclei. So this whole entire thing is the central cell. And in the middle of the central cell, you have the polar nuclei. Two of them are clear. So they say that the mature embryo sac of the angiosperms, it is 7-celled, 8-nucleated. Do you understand this? 7-celled, 8-nucleated. What does this mean?
Because the central cell has polar nuclei, so it will be 7-celled, but it's 8-nucleated.
How is it 8-nucleated? Explain.
Because there's 8 nuclei.
Very good. There are 8 nuclei. 1, 2, 3, 4, 5, 6, 7, 8. Total 8 nuclei, right? 1, 2, 3, 4, 5, 6, 7, and 8. But how is it 7-celled?
Um, 2 nuclei is there in one, the central cell has polar nuclei.
So this bigger one is the cell number 1. And then this is 2, 3, 4, 5, 6, 7. So all in all, the areas where the cell wall is present, that's 7 in number, but the nuclei are 8 in number. So many a times this can be asked in the question that how is a mature embryo sac 7-celled and 8-nucleated? Clear? Any doubts? No doubts? Oh, all right. Now you can note it down. Yeah, you can switch off your video. Right, comfortable.
Done, ma'am. Done, ma'am. Done, ma'am. I'm done.
Done, Justin?
Ma'am. Done, man.